There we go. So when you look down the microscope, you look for the cells and you kind of pick the ones that you think are right. And what that decision process comes from is a little bit arbitrary. There's a lot of potential for bias in that process. And if any of you have read Thinking Fast and Slow by Daniel Kahneman, who won a Nobel Prize in economics, he has done lots of experiments, and one of his sort of key quotes from his book is that, contrary to the rules of philosophers of science who advise testing hypotheses by trying to refute them, people and scientists quite often seek data that are likely to be compatible with the beliefs they currently hold. So you have a strong idea, you're always looking for evidence to try and support your viewpoint. And as scientists, we are somewhat guilty of that. So I feel like a good research question and a direction that many sort of technologists can follow is how can we make the tools and methodology of science so it makes it harder for our biases to influence our research. And in the context of microscopy, as I've sort of outlined already a little bit, this is how an experiment can basically go wrong. So you've got a slide with a certain treatment that you're interested in, and you are looking down the microscope and you recognize a certain visual phenotype, and you image those cells, and you get a bit of a distribution based on some arbitrary features. And then you have another condition where you think the treatment is potent, and you see something that looks kind of unique, and you image those sort of unique cells, and you get another distribution. But what you would have found if you'd imaged all of the cells on the slide, the whole population, you would find potentially that the population was the same. But because of the choices you made, it appears that there is a difference between them. And this is very common in science. People are doing this all of the time, it would seem. So one of the ways around that is that we could coarsely, very quickly, sample everything on the slide. So this is what I mean by the grey squares here. And then on top of that, we could randomly choose from that distribution the cells we want to image in higher resolution with more sensitivity. And another way of doing it is we could coarsely image all of the cells, and then within a certain predefined range, like essentially in a reasonable range of intensity, we could image those. But then there's still a narrative for how we did the experiment. So when we do our publications, we can show this underlying distribution and then tell we could write down and explain why we chose this particular range of cells and justify it. And this would form a narrative for future scientists to question your methodology and potentially look at a different part of the population or another aspect of it that they found interesting. But the problem at the moment in science is this part of the acquisition is very poorly explained, and there's no narrative to really question it in a scientific way. So I'm interested in developing tools that allow us to bring a more quantitative element to this selection of what we're imaging in, for example, super resolution or in time series, where we have less time to actually do the experiment. So you do a coarse kind of pre-imaging phase, and then based on what you see there, you can then choose which cells that you want in a justified way, okay? And then switch to a modality which is more demanding and it takes more time. Make sense? Yeah. So how do we do that? What's the technology behind it? Well, I believe that we shouldn't all buy big screening microscopes for hundreds of thousands of pounds. We should be able to do this with custom microscopes and affordable microscopes. So for example, in my institute we have some state-of-the-art microscopes, and they're very custom. It doesn't make sense to try and turn that specifically, try and put a STED microscope on a screening microscope, for example. What we want to do is be able to do screening on a conventional microscope, so we could still do STED on it because it's using conventional components, but we have the ability to do automation as well. So it's about bringing automation to your regular microscopes. Okay, that's what will enable people to do it, rather than making a super automated STED microscope from scratch, if you see what I mean, a turnkey system. So the way you can automate microscopes is pretty straightforward these days. You can buy automated stages, you can buy high performance cameras, and also a key thing are high performance lamps. These have very strong, these LED lamps that are now available. They're not really lamps, they're light sources. You can switch between the LEDs very efficiently. You can control it via the camera actually, so that it can be synchronized. And it really empowers you to do much more high performance automated microscopy than was previously possible. And then attached to all of this, we have a control system. And in the case of the algorithms I'm using, we have a GPU where we actually run the algorithms. Okay, so this is the equipment that we bought for the project, and I constructed a microscope. You see, I also played around with some of the optics, and we'll talk about that later. And then we wanted a way of localizing the cells which was very fast and also very accurate. And we wanted it to be generic, so that one week we can run one assay, another week we can run another assay. And although in microscopy we've got very good signal processing algorithms, they are what we would define as being somewhat fragile, as in you need a skilled image analyst to tune it for a particular situation, and then that algorithm may work very well, but then you basically have to start from scratch if the visual phenotype of your next assay is different. And if we want to empower biologists, we can't be strictly dependent on the resident image analyst and their particular skills. So we can combat this by using machine vision and computer vision, and there are very powerful algorithms within this field which can be trained to recognize objects in a scene, conventionally in photography. And to train something like this, you just have to do bounding boxes. Okay, so although the design of the algorithm was very elegant and done by real experts, to actually use it, you don't have to be a massive expert. You just have to be able to draw bounding boxes around things that you like. And this class of algorithm is called an object detection algorithm. Okay, in that it allows you to find and localize objects in a scene, and it also allows you to find the class of that object, as in, is it a person, and is it a horse? And you're allowed to have more than one, many classes in one image, which is very powerful. Okay, so the challenge for us was, I know there's a number of leading algorithms out there, but how easy is it to get them to work on microscopy, where we have much less data compared to these very large image data sets? So some of these photography data sets have hundreds of thousands of images in them. In microscopy, you don't want to take 100,000 images before you can run your experiment. So we wanted to know how well we could actually use this technology without having a lot of data. So that was sort of the question. And so I took some of the leading algorithms. We took faster RCNN, we took RetinaNet, we took YOLO. These are leading algorithms within the world of computer vision and photography object detection. And what they all do on some level is and you'll hear more about this today, but what they all do is they use convolutional neural networks as kind of their main building block, where we learn features which will recognize and classify objects. And what we do is, rather than classifying objects specifically, we hijack the rich feature description that these networks generate, and we use it for predicting bounding boxes and for classes. So you're not going to get that from how that really works from this lecture or the explanation, but essentially what it allows you to do is, from this rich feature representation, it allows you to predict and evaluate bounding boxes around your image, and it will give you a probability and a class of that being an object. Okay, and what you do is every kind of bit of this feature will predict in its local environment where objects are, and then you classify what's in those objects, and then you do a kind of maximum suppression just to get one bounding box around each class. And what this allows you to do is potentially, for example, recognize a cat next to a dog and have a nice box that frames it. So it's very very powerful, and it's very quick as well. At inference time, training can take a day or more, but once you've got it trained, it can evaluate what's in an image many times a second, and it can do it with great accuracy. The limitation is, for such speed, is that it has to be a rectangular bounding box or a square, but not a what we call a semantic segmentation, which is on the pixel level, because that's much slower. And if we have a bounding box which is not so regular, then it takes a lot of time to sort them and make sure that they're not overlapping and things like that. So although it is just a rectangle around what we have, it's very fast and it's very powerful for the classification. So those are sort of the limitations of it, but also the reason it works so efficiently. So how does this work with cells? Well, what we did is we took a number of data sets of cells that we acquired on our microscope and also borrowed from other people, and we drew bounding boxes and we trained the network on the bounding boxes. And then here we have some of the example outputs from the algorithm, and you can see it matches our white boxes, what we call a ground truth, very accurately. And this was just trained on 30 images, okay? So that's not very many images in terms of deep learning, but what we did was something called transfer learning, where we pre-trained it on the photography data set, and then we just fine-tuned it on our cellular data set. So really the underlying structure of the network, all of the weights that define how it reacts, have already been learnt. And what we're doing is just fine-tuning it for our objective, which was to recognize these cells. And it turns out, cells is probably similar to footballs or something like that. So it's not such a stretch for the algorithm to transfer what it does from one purpose to another. So I'll go through some performance metrics, but essentially we found very early on that it was performing very well. Okay, but this is a straightforward data set. This is a bit more tricky. This is a data set where we have more than one visual phenotype. It's very important that we recognize all of the cells on the slide. So we want to be able to recognize very diverse appearances in cell. And in this sort of very qualitative example, it does a pretty good job of recognizing the cells, even if they are punctate or if they look like they're filled with fluorescence. Okay, so these are cells expressing peroxisomes, which are very variable. And then we've got things like nuclear pore. So nuclear pore isn't so challenging as the previous example, but most people, when they do DAPI segmentations, the DAPI is very consistent, and this is very dotty. And you can see that even though it's very dotty, it does a very good job of localizing the nuclei. And actually, overall, although this data set only had 26 training images, it does extremely, almost perfectly in terms of localizing the cells in the scene. And then here's another very challenging one with large cells which are overlapping, and again it does a very good job visually. But just showing you examples isn't great because I could have cherry-picked those images. But what we did is we employed this sort of industry standard metric for assessing the localization, something called the mean average precision, which makes sure that your predicted bounding boxes are evaluated in a fair, sensible way. So they have to overlap the ground truth bounding boxes where you've labeled it in your training material, but they also have to not overlap everything. Okay, so the mean average precision basically combines precision and recall metrics into one value, where 1 is perfect and 0 is poor. And I'm not going to go into it in too much detail, but basically, depending on the data set, it will, and depending which algorithm you're using, and since this I've actually tested more algorithms now, more a thorough evaluation. It basically does very well with the most challenging data sets being that peroxisome one I mentioned, and then the neuroblastoma. But if your cells are nuclear pore or DAPI, it basically does it perfectly, which is fantastic, and it's very quick. And I've tried some other things. So there are some things that were interesting that popped out of this. So it turns out that in reality, conventionally, when you do object detection on photography, you do a lot of data augmentation, and I think Mix will talk about this, where you try and amplify how much training data you have by flipping it around and rotating it, creating subtle variations to increase the diversity in what you're using to train your algorithm on. And in photography, you flip things left and right to do that, but you don't tend to flip upside down, because you'd never see in life upside down dogs and upside down cars. So if you train an algorithm to try and recognize those things, it's kind of wasted resource in a way, because it won't see it in reality. But in microscopy, you can flip your input images left and right, up and down, and rotate them how you want, because most conventional microscopy, there isn't polarization in the direction, so you can take advantage of that. And what's this one? Yeah, I'm not going to go through the things, but basically, if you do the flipping, then you get an extra few percent in accuracy, which is nice. And also, what I find is if you train your algorithm to recognize cells across multiple data sets, it will perform better than if you just train it for one specific data set. So what we're doing here is we're creating a corpus of data from which it can try and recognize the cells, and it will do do a better job if you set it the challenge of learning cells across multiple data sets simultaneously and then evaluate it on one of those data sets. So as part of this, I'm creating data sets that you can just use and add to your data as well, so that when you come to train it and use it, you've got this data to help pad out what you've got. So this will add an extra few % as well to the accuracy when you do this. So if you're interested in this, you can check the bioarchive paper. If you just put my surname and bioarchive, then you'll find this paper. And it's the top thing on it's the pinned tweet on my Twitter. Did I mention I have a Twitter account? Is that a yes or a no? Yeah. Okay. So how did I get this to work? Because it's fine just running the images through the algorithm on my computer, but I wanted to control the microscope dynamically. Okay, so I don't want it to image lots of volumes in spaces where there are no cells at all. So what we wanted was an online, a real-time evaluation circuit on the microscope. So the way we did this is we had this high-performance camera which can easily acquire frames higher than the algorithm can handle. And what we do is we acquired the frames on the microscope, and then this goes, the feed goes through Python into our computer vision algorithm, which is running on the GPU, and then we get the bounding boxes, and then this feeds back to our control system, which will then adjust the camera and the rest of the microscope to control the movement and the acquisition. So LabVIEW is this software which can interface with hardware, and it can also keep a Python kernel alive. So the Python's always running and it calls functions from it. So evaluate this image. If you had to load Python afresh every time you wanted to evaluate an image, it'd be very slow. So it's very important that you have a control mechanism which can keep the Python kernel alive and feed it the data dynamically. Okay, and that's kind of why I was using LabVIEW. The reality is now the latest version works completely in Python. So you don't need LabVIEW anymore because people have managed to write the drivers completely in Python. So it makes life a lot easier. Has anyone here used LabVIEW? I feel your pain. I feel your pain. It's great, but it's also an absolute pain sometimes. So this is all good. Any questions so far? No? All good. So this is kind of a tricky bit for a long time, was getting this all to work, but now it does. And here is an evaluation presented on the computer. So this is what my screen would be seeing as I move around the stage, and you'll see me moving it around. And you can see in real time it predicts the bounding locations of the cells. And then this is me doing it manually, but the microscope can be controlled completely autonomously, and it will move around. If it sees a cell, it will then image a stack which just encompasses that cell, and then those cells, and it'll move on to the next area. Okay. But this technology means it knows where the cells are rather than just seeing an image. Okay. And then what you do is basically you get a lot of detections across a slide, but these detections aren't linked. So it sees one image and it sees the cells, it goes down the next level and it sees more cells. Although they're clearly overlapping, the algorithm by default doesn't know that the detections are the same cell. So there has to be a process of linking these detections up and making the volumetric bounding regions coherent. So what I use for this is an algorithm called sort, which is generally used for tracking people as well. And this is a Kahneman filter based approach. And in this example I'm about to show you is a simulation of me putting Gaussians around, and the idea is that the detections are visualized as being coherent if the color of the box remains the same as I move around the image. So if I play this movie, you can see that they stay magenta or green or blue, which means even though I'm moving things around and going in and out of focus, it's able to track them and keep them, identify that it's the same cell from frame to frame. You see, so it's not just detections, we have tracked objects now. And the result of that is now when we do the slides, we have nice bounding boxes with one bounding volume for each cell. So this is a 3D acquisition, and we basically linked up those bounding boxes. So the evaluation isn't done in 3D, it's done on single images, because that's the convention for microscopy, is that you do your reconstruction later. And it's much easier to train if you're just doing 2D bounding boxes rather than doing 3D. And then we link them afterwards, and this seems to work pretty well. Okay. And so once you've got the bounding box, then you can generate an intensity distribution, and this is the foundation of how you can start to get a picture of the whole distribution of your cells that are there and what's going on. So that's great, and in the paper there's actually more about that, and I'm about to actually re-release the preprint, and we're in the process of submission, so we've done some screening on it now, and it's very interesting what you see. It seems to work. But we have to keep the human in the picture, and I picked the most passive-aggressive human and robot picture I could find on the internet. This guy I find quite intimidating, and this robot's in trouble, basically. So the way that I really wanted to do that was to use augmented reality. So who knows what augmented reality is? A few. So you might have seen on a Microsoft HoloLens. This is where you like wear glasses, and then as you look around your environment, you see your environment. It's not a digital environment, but on that environment is overlaid graphics. So you may be looking around a table, you see the table, but there's like an avatar or something in the middle which you can move around. And Microsoft always show it like a ballerina doing a spin on a table. And you can play computer games with your friends with the augmented reality glasses on. So this is discrete from virtual reality, where everything is completely virtual. And I've been telling people Star Wars stories throughout the workshop, and one of them was my virtual reality experience in Westfield Shopping Centre, where after the virtual reality experience, me and my brother still felt like we were in the Matrix. People were looking at us, and it felt like we were somehow alien back in reality. Maybe I never came back to reality, we do not know. But that's virtual reality, but this is augmented reality. So what we did is we designed a system whereby we can overlay graphics as we're looking down the objective of the microscope. So you're seeing your sample like on a normal microscope, but what you're seeing is annotated with graphics. So we're not looking at a computer screen, we're actually seeing our sample, and a screen is being overlaid like a ghost image as we look down. So I'm not the first to publish this, unfortunately. Google beat me to it, but when I wrote the grant, they hadn't done this. That's just my disclaimer. But I'm the first to do it for fluorescence microscopy, and my system's very easy to implement. And basically what I did is I got the engineers at Oxford to build me some custom components and all the blueprints are online. And what there is in the optical path, there's a 50-50 beam splitter here, and perpendicular to that there's a screen, just a regular HDMI screen, and the graphics we project here will appear overlaying the light as it comes off from the sample. So this is very cheap, very easy augmented reality. The newer version won't work like this. We'll actually use transparent OLEDs, which is much more compact, but this is a very easy way of overlaying graphics into your system. And we also engineered a beam splitter by the camera, so the camera gets half the photons come up to the eyepiece, and then this is overlaid as you look down the eyepiece. And then this is actually me with my iPhone pretending to be a viewer looking down the binocular. And you can see it's a bit slower than when you look at it on the computer, but you can see that the bounding boxes move with the sample pretty quickly. So this is the idea is to keep the human very much in the loop and do real-time analysis, so you could actually inform yourself and make decisions about what's going on, and things like that. And another way is, this is still a bit buggy, but rather than just showing the bounding boxes, we want to give a metric for the intensity of these bounding boxes. So we change the shade of the bounding box to represent how bright that cell was. But it's just the start, really. For the remainder of my fellowship and my next grant, I want to build a whole suite of real-time analysis so that you can be informed about what's going on in your microscope just as you're looking around your sample. Okay, so not just for automation, but also just for, oh, I want to know how many foci there are approximately and things like that. I don't want this to enhance people's bias, so it has to be connected to the automation, but we really want to have real-time feedback. Yeah, you had a question.
How does your performance of your deep learning scale with magnification? So if you withdrew, that give you a much worse pixel definition.
Yeah.
Does it fall off?
This is what's I was asked by the reviewers as well, and these algorithms are very. Is it you? Is it you? And these algorithms, these computer vision algorithms, are very good at being scale invariant because they're used to photography where people can appear very close to the camera or very far away. So doing the training, you actually change the scale of your image. So I haven't got anything to show you about how well it works, but it's very good at going between. From what I've seen, it's very good going between different microscopes and different objectives. There are limits to that, of course, but you could easily go from 2 different systems, which were wide field, and it still worked pretty well. And you could go, I don't really know the extent of the range, but you could go from 100 times to 63 times. And it depends where you train it. Like I wouldn't go from 5 times to 100 times and expect particularly good results, but it would work to a certain extent. But you could definitely go from 20 to say 40 or 20 to 10, and it would work. So they're remarkably effective at handling those situations, much better than a specific algorithm written by an image analyst would be. That's the power of them.
Do you define limitation features that would be deeper in terms of shape or in terms of complexity of shape?
Do I define the features?
Yeah, I mean, to what extent you would say deep learning can learn to recognize features? I mean, depending on the feature itself. I mean, the complexity of it. Like if you ever thought about it or tested it.
It can handle remarkable things. I'd say their ability to abstractly recognize features is better than a human. You can't explain some of the things it's able to recognize mathematically, for example, and that is part of the amazing thing about deep learning is that you don't anymore have to choose the features yourself. This is more shallow deep learning, shallow machine learning, conventional approach, which is that you would choose your features and then you'd learn to classify based on that very rich numerical description. Now with deep learning, you go straight from the raw image to what you're trying to do, and it learns everything in between. And although there is some, there's aspects of these features that are learned that you can recognize from a conventional viewpoint, the real beauty of them is that they are non-linear, and they are able to combine information and utilize it across multiple levels. So it's hard to cognitively really break that down and explain step by step how it works, but it does make sense because a lot of people don't really think about what they're doing, but they still function in the world, right? People can't explain their actions. But Mix will spend the day talking about that sort of stuff. But I was so happy at how well it worked. And the real risk was, did I have enough data to train it? But it seems like the objective that I'm trying to do is very similar to photography, more similar than it would seemingly appear to be. So without too much training material, and obviously if it's very challenging data, if the cells are overlapping a lot and it's very variable cells, then you need more training data. But for cellular detection with like DAPI or something like that, it's pretty much perfect. And from that point, you can do different things to sample more complex cells and things like that. So it's a great starting point, and we can do a lot with this, even if there's still some work to go with training on next to nothing, which a human can do, right? It doesn't take much for a human to recognize something. So one-shot learning is kind of the future of these algorithms, I guess, but there's quite a lot to do for us to get to that point. We don't know how to do that yet. Sure, but I'll just finish up. So in summary, I found an excellent way using object detection algorithms for cellular identification. We've benchmarked it in other algorithms on actual data. Surprise is that it works well without too much data. Have enhanced the training and justified why we need a corpus of data to help other people. Can distinguish cells and images can be used to acquire 3D volumes of cells from this 2D localizations. Have developed a real-time acquisition setting system for previewing and screening, and now the next steps. So at the moment we spent $4,000 on our computer, but these days you can actually get what they call edge devices. NVIDIA do them, for example, where you have a GPU on a small board like a Raspberry Pi, a small microcomputer, and these have remarkable powers for doing the prediction side of the algorithm. So we can actually control a whole microscope from a computer that costs about £100 now, rather than using the £4,000 one. We can do it for about £100, and that will be a Linux-based Python control algorithm, and that will, you'll be able to set up using our GitHub account very easily and run your own microscope with it. The only problem with it is that it is an ARM-based chip, which means that some of the hardware drivers that you have on a PC won't work on it, so you have to be quite selective with which cameras and which stages you buy if you want to use it. It's not a plug-and-play system, but if you're willing to, if you design your system now, then you could make it fully compatible with this system and have an automated control system for £100 or something like that. So we're democratizing the technique as much as possible, and you can use it for screening your cells, but you can also will be able to use it for potentially recognizing cells that are going through a certain cell cycle or something like that, and do imaging pipelines that react dynamically to certain appearances of your cell. So you could catch a cell just before it's about to divide and then switch to a time series mode and image that particular location. So we're basically allowing you to automate what you would do as a poor scientist sitting in a cold laboratory doing experiments for many hours, So, but also still trying to keep it fun with the augmented reality and giving you feedback. Sure. So I'll just finish. And some of you have, I've been telling you about my puzzle, some of you, that I've been doing in my hotel room. And I'd just like to thank my previous supervisor, Christian Egeling, and my collaborators that have helped me with the samples, and to the Wolfram Imaging Centre in Oxford, who I'm very much associated with. And yes, thank you very much. Thank you.